So, dear students, very good morning to you all. I welcome you all into the industrial pharmacy first. So, as I said that we are studying the pharmaceutical aerosols since our last three lectures, and in the last lecture we had gone through the different aspects like say the valve and actuators, aerosol system, different types of aerosol systems, and the process by which we can go for manufacturing of the aerosols. Today we will going to discuss in the coming slides, and after going through these slides you will be able to understand, you will be able to know about the most common types of the aerosol inhalers. Then you will be able to understand the process of evaluation of the aerosols, and this will be the very last lecture associated with the pharmaceutical aerosols. In order to achieve all these pharmaceutical aerosol objectives, we have to go through our lesson plan like types of aerosol inhalers and the different kinds of evaluations for the aerosols. So let's start with the types of aerosol inhalers. Now, if we will see, there are major three different types of inhalers. These are termed as MDIs, SVN, and DPIs. MDIs, these are nothing but the metered dose inhalers. SVN, that will stands for the small volume nebulizers, and the DPIs, that will stands for the dry powder inhalers. Now, just from the name itself, we can have an idea that the metered dose inhaler that is responsible for delivering a metered or a measured amount of dose. Small volume nebulizers, these are going to be intended for their use based on their use, their delivery of content that is in the form of a very small volume. And there are dry powder inhalers. That means these are the product which will contain dry powder inside of these inhalers. Now, if we want to see the basic components in case of MDI, that is the metered dose inhaler. So this is the just overview of a metered dose inhaler. You can see there are different components, like say there is a inverted canister, there is a metering valve, there is a expansion chamber, and there is a actuator nozzle. As far as the internal content of this canister, inverted canister is concerned, it will consist of gas phase formulation, and there is a special assembly that is said to be the retaining cap or the retaining cup, and there is a metering chamber. This metering chamber is responsible for measuring the required amount of dose of the content that is to be delivered. So you can see the different parts. These are the canister, metering valve, metering chamber, and the expansion chamber. Generally, the volume of 25 microliter to 100 microliter, in this particular range, the fixed volume is delivered in the form of the meter dose inhalers. And the dose is filled in metering chamber is delivered, and it is based on the metering chamber, it is based on the measuring devices. The inverted canister with the valve below to it, and this in turn assists the gravitational refilling of the valve. So because of the inverted nature of this canister, the process, that is the gravitational pull, that will carry out the process of filling of the metering chamber. As far as the small volume nebulizers are concerned, the devices which deliver the solution and suspension in the aerosol form, these are capable to be inhaled in the lower respiratory tract. You can see it is the small volume nebulizer, volume that is associated with the liquids. So that's why these devices, they will deliver either solution or the suspensions. Now, there are three different types of the small volume nebulizers, that is the pneumatic jet nebulizers, ultrasonic nebulizers, and the mesh nebulizers. So, the pneumatic jet nebulizer, that is, it is associated with the liquid jet. Ultrasonic, that is the different waves or the electric current that is used, and the mesh, that is nothing but there is a screen, there is a mesh which is responsible for delivering the particles of a required size. Pneumatic jet nebulizer, pneumatic that means the liquid based, and the jet that is nothing but the pressurized airflow. So this is the pneumatic air jet system. You can see there is a compressed gas source which is attached to this particular device. There is a liquid reservoir, there is a dead volume, and there is a baffle orifice. There is ambient air and drug loss during the exhalation. So the required amount of air that is needed to be provided, and from the another side, it is to be held by the patient, and then it is inhaled. The pneumatic jet nebulizers, these are responsible for the delivery of compressed gas through a jet, and this will entrain the product into the compressed gas. So this gas is responsible for carrying. This gas is responsible for expelling. This gas is responsible for dispersing the product. The product is shared into liquid film and the unstable film that will breaks into the aerosol droplets. So that is under the action, that is because of the effect of the compressed gas. It will carry out the process of releasing and sharing and breaking the aerosol in the form of the droplet, the product in the form of the droplets. There is an ultrasonic nebulizer. In case of ultrasonic nebulizer, the electricity is used in order to generate the ultrasonic waves, and it converts a liquid into aerosols which can respired or inhaled. You can say there is a drug solution, and this drug solution that is in association with the piezoelectric transducer. This piezoelectric transducer that is in connection with that of the power transmitter. When this power is provided, at that time the waves are generated, and these waves are responsible for creating the aerosols, and these are going to be inhaled or respired. There is a mesh nebulizer. Now this mesh nebulizer that will involve the use of a mesh or a plate with multiple aperture to produce a liquid aerosol. Now this can be compared just by comparing it with that of the device which is going to be used in case of garden for giving water to that of the plants, right? So as it will consist of a sieve and the device that is responsible for dispersing the water into the form of the fine droplet. So in the same way, this device is there, which is said to be the mesh nebulizer. There are the dry powder inhalers. So all what we saw up till now, these are the examples of the nebulizer. That is the mesh nebulizer, ultrasonic nebulizer, and the pneumatic jet nebulizers, right? Small volume nebulizers. Then we saw the MDI. Now we have to move to the DPIs. So DPI, that is the third kind of inhaler system, and it is the dry powder inhaler. So as it is the dry powder inhaler, the powder that is to be placed inside this, and it is propellant free, and it is breath actuated. Now it is propellant free. That means no propellant is going to be involved. It is going to be added because of the deleterious and the environmental hazardous effect of the CFCs, like say ozone depletion and producing the greenhouse effect. and increasing the global warming. So under the Montreal Protocol, so these devices are developed and they design post Montreal Protocol, and these are said to be the post Montreal Protocol inhalers, and which do not contain or which do not go for use of the propellants. So that's why these are the propellant free. Then how to carry, generally these inhalers, these are going to be used in case of treatment of lower respiratory tract, like say the lungs, right? Now how to carry the content or the powder of the therapeutic activity till the lungs. So in order to carry it, there is a breath activation system. That means when we are using is, at that time we have to inhale. So along with the inhaled air, the powder particle that will goes inside of the lungs. So the examples are the dry powder inhalers, that is in case of insulin delivery, like say the various commercially respiratory sprays of insulin that have been approved by the US FDA. The examples are the Exvera and the Arax, etc. So you can go for finding few more examples of the insulin inhalers. Now, what we had studied up till now, or we saw, let us summarize these things. We saw the most common inhalers. What are these? These are the MDI, meter dose inhaler, SVN, small volume nebulizer, and the DPI. DPI is nothing but the dry powder inhaler. MDIs, they will contain the different part, like say inverted canister, metering valve, metering chamber, and the expansion chamber. Small volume nebulizer, these are of three different types: pneumatic, ultrasonic, and the mesh nebulizers. Pneumatic, that will involve the use of the liquid and the air system. Ultrasonic, the electric current is used. In case of mesh nebulizer, the sieve or the mesh or the aperture device is used. In case of dry powder inhalers, these are the portable, these are propellant breath activated inhalers. So this is most important, propellant, because generally these DPIs are used in case of the asthmatic patients. Asthmatic patient, hardly it is very difficult to carry anything with him. So that's why these devices are designed in such a way that these are light in weight. So these are small in size so that they can be carried with them. And now we will go for studying the process of evaluation of the aerosols. As far as the evaluation of the aerosols are concerned, there are four different tests or the ways by which we can go for evaluating. The first, that is the flammability and combustibility. Second is the physicochemical characteristics. Third is the performance, and the fourth is the therapeutic activity. As far as flammability and combustibility is concerned, there are two different tests: flame projection and the flash point. In case of flame projection, generally the effect of an aerosol on the extension of an open flame after exposure of spray for 4 seconds in flame, that is to be observed. So that is the flame projection test. How long, for how much duration the flame that will going to be carried. Here you can see it is just for the 30 centimeter, right? So we have to go for spraying, and this is generally in case of the continuous spray systems, and the spray has to be kept actuated in case of the 4 seconds or for a duration of 4 seconds. Then there is a flame projection flash point test. Now this flash point test, that is the test which is to be performed in case of an apparatus, that is the USB apparatus. So that is said to be the tag open cap apparatus. What it is? It is the tag open cap apparatus. Keep in your mind, this is the apparatus which is used in order to perform the flash point test in case of aerosols. So in order to perform this test, you can see this is the apparatus. This is the tag open cap apparatus. Tag open cap apparatus. The inhaler or the aerosol has to be placed inside this. But before placing it, it is going to be chilled to minus 25 degrees centigrade Fahrenheit. Then it is transferred to the test apparatus and slowly increase into the temperature that is to be done. The vapor ignition temperature, that is noted as a flash point. So the vapor, they will get ignited. So that temperature is going to be noted as a flash point test. Next is the physicochemical characteristic. In case of physicochemical characteristic, the aerosols, they has to be evaluated for the various parameter like say the vapor pressure, density, moisture content, and the identification of the propellant system. In case of vapor pressure measurement and evaluation is concerned, generally the pressure gauge is used. In order to measure the density, the hydrometer or the pycnometer is used. To determine the moisture content, the Carl-Fisher method is adopted. And in case of identification of the propellants, the aerospectroscopy that is to be used. Next is the performance. So the performance that is in the form of the aerosol wall discharge rate, the delivery rate, the delivered dose uniformity, net content, minimum fill, leakage, and the particle size distribution. So these tests are to be performed in order to evaluate the aerosols. So aerosol wall discharge rate and the delivery rate, it is in accordance with that of the USP. So this is to be determined by taking an aerosol of known weight, then discharging the content for a given time using the standard apertures, and by reweighing the content after time limit has been expired. So the change in weight per time dispensed is the discharge rate, and it is expressed as the gram per second. Note that it is to be expressed as the gram per second. How much gram of aerosol that is delivered in a given second. Then the delivered dose uniformity, that is how much amount of dose that is going to be delivered in per actuation. To determine the amount of medication actually received by the patient, evaluation of dose uniformity test is done. The method that will enhance the accurate weighing of the filled content followed by dispensing of the several doses. The container is again reweighed and the difference in weight divided by the number of doses that will gives the average dosage. Acceptance criteria. In case of nasal spray, the USP has given the acceptance criteria. Not more than 2 of the 20 doses are outside the range of 80 to 120 of labeled claim, and none are outside the range of 75 to 125 of the labeled claim. So whatever the claim has been made, all they should remain between 75% to 125%, right? No one should go outside. And if we are going for the 20 doses, out of these 20, if two they are remaining outside of the 80 and 120, but if these are less than or between the 75 to 125, then we can say this is the acceptance criteria. While the mean for each of the beginning and the end dose that should falls within the range of 85 to 115 of the label claim. So the mean should not go outside the 85 to 115%. Then the another acceptance criteria, let us say for example, if 3 to 6 doses of the 20 doses collected, these are outside the 80 to 120, right? So this is in accordance with that of the first. If two are outside 80 to 120 and between the 75 to 125, and if the average is between 85 to 115, then it will accept or it will passes. But suppose out of these 20, if 3 to 5 these are outside the 80 to 120 of the label claim as it has been shown here, but none is outside the 75 to 125% of the label claim, and the mean of each of the beginning and the end doses falls within the 85 to 115% of the label claim, then you need to go for selecting the additional 20 containers for the second tier testing. Now, in case of the second tier testing, for second tier testing, the requirements are met if more than 6 of the 60 doses, we have to go for the 60 dosing. If more than 6, that means 10%. If more than 6 of the 60 doses collected are outside the range of 80 to 120 of the label claim, but none is outside the range of 75 to 125% of the label claim, and the means for each of the beginning and the end doses fall within the range of 85 to 115% of the label claim, then we can say it will pass as the test. So that is the second tier testing. Now the USP is very, very specific as far as the process of evaluations are concerned, and they are having the set standards. Like say for example, they recommend that we should go for use of a mechanical means of activating to deliver the dose for collection, because say the actuation force, actuation speed, actuation stroke length, rest periods, like say parameters, these must be controlled, because say from person to person these will vary. Person to person actuation force is different, speed is different, the length is different, and the rest period is different. So there is a need to have this set standard procedure. The test must be performed on units that have been primed according to the patient use instruction. So what instructions has been given into the leaflet, in accordance to that it should be carried out. For suspension product, the delivery dose should be delivered into the scintillation vial, the special types of the vials that are used in order to carry out the process of evaluation. Now the net content and the minimum fill, that is how much amount of product or how much that particular aerosol system is filled. So the wet filled full container, after removing labels and cleansing outside surface, that is number 10. 10 number of container has to be selected. Their labels has to be removed. They has to be cleaned properly from the outer surface, and they should be weighed. The full container that is to be weighed. Then remove all the content. The content has to be removed. Then they has to be heated at 100 degrees Celsius for 5 minutes. Then reweighed all the container individually. Why it is responsible or why it is necessary to heat them up to 100 degrees Celsius for 5 minutes? Because the volatile content, volatile propellants are there, so that may adhere to that one. So the difference in weight, that will be the net fill weight, right? So the filled container minus empty container, that is the actual filled weight for this. The acceptance criteria, that should be in accordance with that of the USP, and the requirements that are met if the net weight of the content of each of the 10 container is not less than the labeled amount. What amount has been labeled, it should be in accordance to that one. Then only it will be accepted. It will passes the test. The last test, that is the leakage test, and this is to be performed at last. No doubt this is going to be carried out during the process of manufacturing itself, but this is to be performed because the test is used in order to estimate the weight loss over a period of the 1 year period. So for a period of 1 year, if the product that is going to be considered as a during its shelf life, so there should not be any kind of leaking, and if leaking is there, that means the product that will loss. And this is to be to perform this test only on topical aerosols fitted with continuous wall. The leakage test, that is to be carried out only in case of the topical aerosol systems, and that too with the continuous wall, not with the metered wall. This test is in addition to the customary inline leak testing of each container. During production itself, the leakage test that is to be carried out. So from product to product, the leakage test that will differ, and it is given in case of the USP. In accordance to that, they should be followed. Then there is a particle size distribution. This is of great importance in case of more specifically the suspensions, because the product that is going to be inhaled, more specifically in case of the dry powder inhalers, the powder that will goes inside of the lungs, and from the alveoli it is getting absorbed in order to show its therapeutic action. So that's why the particle size that is most important one. The particle size is generally evaluated by using the cascade impactor. See, all the test or the description is there that you have to imagine. There is a system or this particle size distribution in case of pharmaceutical aerosol. This can be evaluated by using which particular apertures. It is the cascade impactor, right? So sometimes this may be asked into the MCQs. So in these apertures, the particles are carried in a stream of air through a series of consecutively smaller jet opening. So there are consecutive layers, and through these layers, there is a stream of air, and these will passes of a smaller jet opening. The first will contain higher, second higher, smaller, third smaller, fourth smaller, likewise in the decreasing fashion. The heavier and larger diameter particle are impacted on a slide under the larger opening. As the opening gets smaller, the velocity of the stream increases and the next larger particle are deposited on the next slide. So as there are different slides, the heavier particles, because they cannot run for a longer distance, so they will be impacted to the first slides in accordance to their weight. The one whose particle size is smaller, they can run for a longer distance and they can impact to the lag, to the slide which is located at a longer distance. The therapeutic activity. Now, this is the by standard in vivo test, and it is as per the pharmacological models. It is to be performed in order to evaluate the therapeutic activity and the efficacy of the aerosol systems. So this is used for assessing the inhalers, specially designed insufflators, and are used to deliver the powder or the solutions to the test animals. The gamma scintigraphy and the single positron emission computer aided tomography imaging techniques are going to be used in order to perform the therapeutic activity of the pharmaceutical aerosols. Which equipments are used? The gamma scintigraphy and the single positron emission computer aided tomography. And for topical sprays, normally these are applied onto the test animals. And dear students, so with this, we had finished the process of evaluation of the aerosols. Thank you very much. Have a good day to you all.